Hello, folks. Welcome to the second lesson of Unit 1 for the Advanced Higher Biology course. So, Topic 2 is all about proteins. So, we're going to be looking at the function of proteins, how they are produced, and a little bit about their structure and how they interact and how that structure is related to their function as well. So there's quite a lot to get through in this unit, this topic. Sorry, it's one of the longest within the course, and there's a whole range of things to cover. So what we'll probably do is split this video into two, and we will cover the first half, and then we'll have a break and we'll do another video for the second half. So first of all, we kick off by looking at proteomics. Now, proteomics is the study of the entire expression of proteins from our genome. Okay, so we know that our genes are expressed to produce proteins characteristic for a particular cell type. We know that from higher. The proteome is the entire set of proteins that are expressed by the genome. And we can see this nice little diagram here shows us the relationship there where, the DNA obviously becomes RNA, and we get our maturing primary transcripts and so on from the messenger RNA. And then that goes on to produce all of the different proteins. Now, we know from higher that one gene on our DNA can code for many proteins. And that's how we're able to produce such a vast number of proteins from just a relatively few number of genes. So we have about 25,000 genes or thereabouts that have been discovered, and we have over 1 million proteins to be produced there. So that means we need lots of variants through alternative RNA splicing to produce all of those different proteins. So the proteome is larger than the number of genes, particularly in eukaryotes like ourselves, because more than one protein can be produced from a single gene as a result of the alternative RNA splicing, as we just discussed. But not all genes are expressed as proteins in a particular cell type, okay? We have specific proteins that are produced for that particular cell type, okay? And we learned about that in higher human biology, where we looked at the proteins that are expressed through cellular differentiation are characteristic for that particular cell type. So genes that do not code for proteins are called non-coding RNA. And those non-coding RNA genes include those that are transcribed to produce tRNA, rRNA, and RNA molecules that control the expression of other genes, okay? And we'll have come across T and rRNA in higher, and the other RNA molecules that control the expression of other genes within our genome. The set of proteins expressed by a given cell type can vary over time and under different conditions. So some of the factors that affect the set of proteins that are expressed by a given cell type are the metabolic activity of the cell. Okay, so how quickly all the reactions are happening. That's our metabolism, the sum total of all the cellular reactions within our cells. So the metabolic activity will dictate that. Any cellular stress, okay, so depending on what stressors are impacting the cell, that will impact the proteins that are produced. Its response to signaling molecules, okay, and we'll learn a lot more about signaling molecules later in the course, and diseased versus healthy cells. So obviously, if a cell is diseased, that's going to affect the proteins that are produced versus healthy cells. And just to give a quick example, for example, a diseased cell will likely be producing antibodies or an equivalent protein to manage that, whereas a healthy cell might be only producing proteins that are required for the functional cell, when it's operating as it should. So it will vary depending on the state of the cell. Okay, and these are some of the factors that will affect the set of proteins that are expressed by any given cell type. So I'm showing this video on the screen here. We're not actually going to watch this video as part of the video. We're not doing a video within a video within a video sort of thing. Starts turning out like the movie Inception. But we are. I'm suggesting that maybe you go and look this one up on YouTube. Okay, so it's called The Structures and Protein Synthesis, and it has this thumbnail on it, and it's really good for allowing you to visualize some of the structures that we're going to learn about next. So we are going to be learning next about the synthesis and transport of proteins. And in order to understand the synthesis and transport of proteins, we need to know all about some of the organelles that we have maybe not learned about so far in National 5 and then higher biology. So first of all, we kick off by talking about intracellular membranes. Now, Intracellular membranes are so important for a cell because eukaryotic cells have a system of these which increases the total area of membrane. Now, you might be thinking, well, why is it important to have a huge area of membrane within the cell? Well, we have a plasma membrane on the outside, but because of the size of the cell, eukaryotic cells have a relatively small surface area to volume ratio. So by having more membrane within the cell surrounding many of the cell structures, that improves that ratio. Okay, it gives us a greater total surface area of membrane. And that's good because many of the important cellular reactions and cellular activities take place on the surface of membranes. Okay, so let's look at some of the structures involved in the structures that have membranes within the cell. So the first one to talk about is the endoplasmic reticulum, okay? So the endoplasmic reticulum, the ER. Now, if we look at this diagram here, we can see there are two types of ER, okay? All of this red and then this sort of orangey area here, all of that is our endoplasmic reticulum. And you'll notice that it's actually physically attached to our nuclear membrane. That's the membrane that surrounds the nucleus there. Okay, so the ER, the endoplasmic reticulum, is continuous with the nuclear membrane. And we can see if we follow the path out from the membrane, we can see that series of membrane tubules which form the endoplasmic reticulum, and we can follow them along. It's a bit like a labyrinth, following all these little tubules along, following the pathway through it. Okay, so the endoplasmic reticulum plays an important role for the synthesis and transport of proteins, and we'll talk more about that in a few moments. Another structure that's important in this process is the Golgi apparatus, okay? And that's a series of these flattened membrane discs. Now, in this bottom diagram here, we can see the Golgi apparatus and we can see that series of flattened membrane discs. Now, this is going to play a vital role in the modification and the finalization of proteins, starting to move them towards their final conformation, their final shape. So we'll talk more about the Golgi again in a few moments, but the ability to identify these structures is important for the exam. So that's why we're going through where each of them are and what they look like first of all. We've also got lysosomes within the cell. Now, lysosomes are membrane-bound organelles that contain a variety of hydrolases that digest some of the main molecules within the cell, such as proteins, lipids, nucleic acids, and carbohydrates. Okay, so they have an enzyme or a series of enzymes called hydrolases within them, and those hydrolases digest all of our main macromolecules there. Okay, so lysosomes are membrane-bound organelles, and again, we can see them here in this bottom diagram. This green structure here is a lysosome, packed with these enzymes that will digest and break down some of our major molecules here. We also have vesicles. Vesicles transport materials between membrane compartments. And I always say to my class, the easy way to remember that is that a vesicle is the vehicle of the cell. A vehicle transports people or it transports things around. And a vesicle does the same job, but within the cell. Okay. So a vesicle is the vehicle of the cell. It transports materials between the various membrane compartments. And then lastly, we've got the cytosol. Now, the cytosol is the fluid part of the cytoplasm, okay? Sol meaning like liquid or fluid, generally in science. So cytosol is the fluid part of the cytoplasm, okay? And we can see the cytosol here surrounding all of our organelles within the cell. So lipids and proteins are synthesized in our endoplasmic reticulum up here, okay? Lipids and proteins, but each are synthesized in different parts of the endoplasmic reticulum. So we have to know the two different parts. First of all, we have the rough ER or the RER, okay, which has ribosomes on its cytosolic face. Okay, it has ribosomes because it's going to produce proteins. And when we think about that term cytosolic, all that means is that it's the face of the membrane, the side of the membrane that's facing the cytosol. Okay, sounds like a complex term, but that's all it means. So rough ER has ribosomes on its cytosolic face. And it has those ribosomes because they are going to synthesize proteins, but we'll talk more about them in a few moments. While the smooth ER or the SER lacks ribosomes. And we can see that in the diagram, the smooth ER there with nothing on it, and the rough ER with all those ribosomes attached to the cytosolic face. In the smooth ER, we produce lipids. Okay, so lipids are synthesized within the SER, and they then get inserted into the membrane of the SER. Okay, and then what they'll do, we'll see another diagram in a few moments that will show the journey of the lipids once they are synthesized and inserted into the smooth ER. Okay, so the synthesis of all proteins begins in the cytosolic ribosomes. Okay, now this is probably what we would expect based on our previous knowledge of translation, for example, at higher level. Ribosomes are found within the cytoplasm and the cytosol, and they will be the starting point for the synthesis of all proteins. Now, if the protein is going to remain a cytosolic protein, okay, then it will complete translation in the ribosome in the cytosol. So if the ribosome is producing a cytosolic protein, then the synthesis of that protein will be completed within the cytosol. And that means that these proteins will remain in the cytosol. So if you're producing a protein that's to remain in the cytosol, the ribosome remains there too, and it completes its job of synthesizing the protein there. However, some of our ribosomes will actually move away from the cytosol after translation has started. Now, what tells the ribosome whether it should complete translation in the cytosol or whether it should complete it somewhere else? Well, there is a short signal sequence. Okay, so that's a short stretch of amino acids at one end of the polypeptide. Now, we remember from higher we had start and stop codons, which a ribosome could use to indicate whether to start and stop producing our polypeptide. Well, this signal sequence is similar in that regard, in that it's a short stretch of amino acids this time. And when we have that short stretch of amino acids being produced and strung together at one end of the polypeptide, that will determine the eventual location of the protein in the cell. So that will instruct the ribosome whether protein synthesis should be continued in the cytosol or whether protein synthesis needs to go elsewhere within the cell. So the eventual location is going to be determined by that short stretch of amino acids called a signal sequence. Now we have, as well as cytosolic proteins, we have another group called transmembrane proteins. Now the transmembrane proteins, as you can probably guess from the name, are eventually going to cross the membrane, okay? Transmembrane. They will cross the membrane eventually and leave the cell. So because of that, they will not remain within the cytosol to complete protein synthesis. The ribosomes that are synthesizing them instead will halt translation at the point of the cytosol, and they will direct themselves towards the ER, and they will dock with the ER to form the rough ER. So those ribosomes that were synthesizing the protein will then dock with the ER to form rough ER. And the way I think of it in my head is I always think ribosome R, so R plus ER equals RER. Okay, the ribosome docking with the endoplasmic reticulum gives us rough endoplasmic reticulum, or RER. R plus ER equals RER. Okay, so if it's a transmembrane protein, it needs to dock with the endoplasmic reticulum to complete protein synthesis. And then once it's docked with the ER, translation will continue. The rest of the polypeptide will be produced. And then that will be inserted into the membrane of the endoplasmic reticulum, and it will pass through that series of membrane tubules that we talked about earlier on. Okay, so the ribosomes in the surface will complete translation. Then the protein or the polypeptide will then insert itself into the rough ER and work its way through these series of membrane tubules. And again, this is a nice point to maybe pause and think, okay, I can't really visualize how this is happening. So what to do is to go onto YouTube, look for this video with this thumbnail, type in synthesizing proteins, look for this thumbnail, and that's the video to watch, because that's quite a nice visualization of how proteins are synthesized in the rough ER, and then how they are then become transmembrane proteins and transported out of the cell. So how does this work? Well, this diagram, I think, shows us nicely what is happening here. So within the ribosomes here, we have these transmembrane proteins being produced, okay? Now, what they'll then do is they'll insert themselves into this rough endoplasmic reticulum, and then they will work their way through the membrane tubules. Now, when they get towards the end of these membrane tubules, they will then be transported by vesicles that bud off, okay? And we can see vesicles here that are budding off the rough endoplasmic reticulum, and they will then move up here and fuse with the Golgi apparatus, okay? And then as the proteins move through the Golgi apparatus, they undergo something called post-translational modification. Okay. Now, post-translational modification involves the addition of it can be fats, it can be carbohydrates, it can be any other sugars, it can be other molecules as well. But the SQA want you to know that the major modification that's going to happen here is the addition of carbohydrate groups. Okay. So when we talk about post-translational modification, there are many modifications that take place, but the major one that the SQA would like you to know about is the addition of carbohydrate groups. Okay. And that comes because enzymes catalyze the addition of various sugars. And we know that sugars are the monomers of the polymer carbohydrates. Okay. So we do that through a series of steps, and eventually we get these polymer carbohydrates. And that's one of the major forms of this post-translational modification that takes place here in the Golgi apparatus within the cell. The molecules will then move through these Golgi discs, okay, that series of flattened discs there, okay, it's going to move from one to the other, okay, and it'll do that by budding off from one disc and then fusing to the next one in the stack, okay, so it will bud off and then it will fuse to the next one in the stack, okay, and try and use that vocabulary when you're describing it in your answers as well. Now, how does it actually transport those proteins through the cell? Well, we've got these vesicles, and we talked earlier on about the vesicles being the vehicles of the cell, okay? They transport things. So the vesicle, and we can see it in this diagram here, the vesicle will leave, it will bud off from the Golgi apparatus, and it will take the proteins which have now been modified, okay? They've gone through post-translational modification, and then it will take these modified proteins through the cytoplasm here towards the membrane, and when it gets to the plasma membrane, it will then fuse with the plasma membrane, and it will then force those components to move out with the cell. So vesicles that leave the Golgi apparatus take the proteins to the plasma membrane, or they can take them to lysosomes. And you can see here the proteins, the enzymes, in all likelihood, the hydrolases that are produced in the Golgi apparatus will then enter our lysosomes here. Okay, so the lysosomes take those proteins, and those will remain within the cell so that they can digest proteins, carbs, nucleic acids, etc. The vesicles, how are they able to move? How is this vesicle able to move from the Golgi to the plasma membrane, and this vesicle able to move from here to the lysosome? Well, it does it through a series of things called microtubules. Now, later on in Unit 1, we'll learn about the cytoskeleton of the cell, and we'll learn a bit more about microtubules. But this little GIF here that I found online is quite a nice representation of how the vesicle is transported, okay? This is the vesicle here, this big sac-shaped thing at the back here, and that will contain our protein. And the way it gets transported through the cell is via this motor protein here, which will bind to the cytoskeleton, to the microtubules, and transport the vesicle from one place to another. So it'll move along those microtubules to the other parts of the cell, the other membranes, such as the lysosome or the plasma membrane, and it will fuse with them within the cell. So that's the key. Again, another important word to use, it fuses with the membranes. Okay, so secreted proteins are translated in ribosomes on the ER and enter it through its lumen. Okay, so that's the lumen in this area here within the rough ER, okay, into these areas here. And then what it will do is it will work its way through this part of the rough ER, and then they eventually arrive at the Golgi apparatus, which we know happens via these vesicles, okay, and we can see it being transported across there. And then eventually when it leaves the Golgi apparatus, it's packed into these secretory vesicles. Now, secretory vesicles are special ones. Those are ones which are going to take the protein from the Golgi to the plasma membrane to leave the cell, okay? We've got other vesicles that remain within the cell, but we've got the secretory vesicles. Those are special ones because they're able to fuse with the plasma membrane, okay? You can see it here, they fuse with the plasma membrane. and they're then able to release the proteins out of the cell. Now, examples of those proteins that might be released out with the cell are things like peptide hormones, something like insulin, for example, or digestive enzymes, so something like amylase or something similar. They will then be taken to the places in the body where those proteins will become useful and active. We don't want them being active within the cell, and that's why many proteins are synthesized as what are called inactive precursors, okay? So they are not in their active form when they're within the cell, and what they require is they require something called proteolytic cleavage, okay, to produce active proteins. So they'll leave the cell, the enzymes will move towards the intestinal tract where it's going to break down whatever it's going to break down, okay? So proteases for protein or lipases for fat or whatever it's going to be. And when they get there, they'll be inactive, but something there will undergo proteolytic cleavage of the protein, and that will activate the protein so that it can carry out its normal function at that site. Okay, so initially they're inactive precursors, but when they get to the site where they want to be active, they'll undergo proteolytic cleavage and will then have active proteins. Okay, now it mentions here proteolytic cleavage is another example of post-translational modification. Post-translational modification simply means any modification that happens to the protein after translation in the ribosome. Okay, so basically everything we do to the protein after that point is post-translational modification. And digestive enzymes are an example here. It mentions of secreted proteins that require that proteolytic cleavage to become active, okay? Because we don't want these digestive enzymes becoming active here in the cell or even near the cell, because we don't want them to be breaking down the proteins or the lipids or the carbohydrates within our cells. We want them to wait until they get to the digestive tract so that they can do it there. So that's the end of part 1 of 1.2 proteins. We'll pick up part 2 and we'll look at structure in the next lesson. Okay, folks, I hope that's been helpful. As usual, leave me a comment of things that you think you've learned from this or things which you could do with more clarification on. But until the next lesson, thanks for joining me, and class dismissed.